My name is Hanieh Noeparast. I'm a PhD student at Western Sydney University School of Science, and I completed my bachelor's degree in microbiology at University of Tehran. And while pursuing my master's degree in food science at University of Illinois Urbana-Champaign, I started my research on CRISPR-Cas-based detection tools for pathogen detection. And building on my background in the food industry and the experience I've got through research, I decided to do my PhD at Western Sydney University in the same field. My PhD focuses on reducing pre-harvest and post-harvest loss of finger lime, which is a high-value Australian native citrus species with significant commercial and export potential. And as demand continues to grow both domestically and internationally, protecting its quality and reducing its post-harvest loss will become very important for growers and for the horticultural industry. Pre-harvest and post-harvest diseases are major challenges for growers, especially fungal pathogens such as Diaportes citri, which causes melanose, and it significantly reduces the fruit marketability. My project has two main sections. The first is improving post-harvest management through packaging systems and storage conditions in order to minimize the disease development and chilling injury at a very low temperature. And the second part involves developing a CRISPR-Cas detection tool for detection of Diaportes citri, the fungus that is causing melanose, at a very early stage before symptoms appear. So in simple terms, CRISPR-Cas12a works like a biological sensor. So it is designed to detect the unique genetic signature of Diaportes citri. So whenever the fungus is present, the system will be activated and a very clear and measurable signal will be produced so that it will allow the infections to be identified at a very early stage. But one of the biggest challenges in this space is that the current detection approaches are usually slow and they are lab-based, and they often only confirm disease once symptoms appear. So by the time farmers can see the problem, losses have already occurred. So my research aims to tackle this by working toward a rapid, sensitive, and field deployable test that can support the early decision making. Research is being carried out under the supervision of Dr. Michelle Mack as my principal supervisor and Dr. Sunil Panchal as my co-supervisor. And I'm also honored to have Professor Paul Halford in my supervisory panel. A very important collaborator on this project is Dr. Frank Beeden at Plant Innovation Center, DAF. I have greatly benefited from his expertise in plant pathology and molecular diagnostics. I also benefited a lot from the expertise of Dr. Matthias Silva Campos at Plant Innovation Center. And I would also like to acknowledge the Queensland Herbarium for providing the confirmed Diaportes citri isolates in order to validate my detection kit. And finally, I'm very grateful to Future Food System for their funding and for their support in promoting the project, because it significantly enhances its visibility and industry engagement. There are two main outcomes that I'm aiming for in this project. The primary outcome is to validate a detection tool, which is based on CRISPR-Cas, which is a molecular tool. And alongside the detection kit, this research will generate supporting evidence on post-harvest handling practices, such as packaging systems and storage conditions, in order to reduce the post-harvest loss of finger lime. So in this way, growers can combine the early detection with appropriate packaging in order to extend the shelf life of finger lime and increase in export standard. And finally, by conducting surveys, including different stakeholders such as distributors and growers, I aim to provide knowledge-based recommendations on handling practices of finger lime. I think the future of food production is very promising because science and sustainability are increasingly working together. And nowadays we've got some advanced technologies like molecular diagnostic tools that they can help reduce disease development and chilling injury, so they will all lead to reduction in post-harvest losses and they will help to food production. And on the other hand, there is growing global awareness around food security and sustainability, and also there is a stronger collaboration between researchers, growers, and industry. So I think that all of this will strengthen the food systems and it will make the food system become resilient and sustainable in the future. Winning the 2025 Future Food System Shark Tank was a very meaningful experience for me. I introduced my research and I shared the vision behind it. And receiving that recognition was incredibly amazing and encouraging to me. It showed me that the value of research lies not only in technical depth, but also in how clearly and confidently we communicated. That experience strengthened my experience in presenting my work to a large group of audience. And to me, it was not just about winning, it was about knowing that the work I'm doing has relevance and impact beyond the lab. After completing my PhD, my main goal is to work in a government sector in the field of biosecurity so that I can contribute to strengthening the early detection systems for plant pathogens. And I would like to work with some technologies such as early detection systems for plant pathogens because it can help Australia's economy, agriculture, and sustainability. I think that science is not only about experiments or publications, it's about learning how to think critically and how to think outside the box and how to solve real-world problems. And research teaches you patience, independence, and perseverance. And it challenges you, but also it would give you the opportunity to contribute in meaningful ways. So there will be challenges and failed experiments or moments of doubts, but they are not sign of weakness. They are part of the process of discovery.